I'm Patrick Danaher, NanoString's Senior Statistician. Welcome to this webinar, in which we'll explore in detail example results from the Pan-Cancer Pathways Analysis module. This is the same dataset that was included with the download of nSolver 2.5 and described in the Pan-Cancer Pathways Analysis module white paper. You can use this data to replicate the analysis in the white paper. The results I show here may differ from the white paper based on different analysis options selected. For all analyses, the software returns extensive descriptive plots useful for exploring your data, understanding its structure, and performing quality control. These plots fall under the QC tab of the report, and they are an excellent place to begin exploring your results. Beginning on the Summary tab, we see two heat maps. On the right, a heat map shows an entire matrix of expression data. Samples are colored by the covariate selected for the analysis. The mapping of covariate values to colors is given elsewhere in the report. On the left, a heat map shows a correlation matrix of the samples. This heat map can provide a better feel for the similarity between samples. Scrolling down, we see a plot showing the mean and variance of all genes in the analysis. High variance genes, which may be particularly biologically interesting, are named. We can also see that our selected housekeepers, in brown, have very low variance overall, and that our discarded housekeepers in yellow have slightly higher variance. It is good practice to understand the effect of every available covariate on gene expression. The plot on the right draws histograms of p-values for the associations between all genes and each covariate. Covariates with no association with gene expression will have flat histograms, and covariates with widespread effects on gene expression will have peaks near zero. Technical covariates with such left-weighted histograms should be adjusted for in downstream analyses so as to avoid confounding. In some cases, a covariate with no effect will be correlated with a covariate with a powerful effect, producing a left-weighted histogram. This phenomenon is likely the case with scan date, whose association with so many genes is most likely a result of its separate association with the covariate subtype and not a real effect present in the data. The histogram for binding density is more likely a real effect. Binding density serves as a surrogate for total RNA input. Samples with low RNA input will be given larger normalization factors, and normalized expression of genes in or near the background of the system will vary with the sample's normalization factor. Thus, binding density will often predict the normalized expression of genes in the background. Finally, and encouragingly, our biological variable of interest, subtype, appears to profoundly influence gene expression. Its histogram is by far the most left-weighted of all of them, indicating that almost every gene in the analysis appears to respond to subtype. Next, we click on the Pair Plots link. These plots examine the basic details of study design, plotting every covariate against every other covariate. Here, subtype is compared to binding density. The two variables have no notable association, meaning binding density will not dramatically confound analyses of subtype. On the top right, a plot shows the number of each subtype at each scanned date. The subtypes are generally well dispersed over the dates, although the normal samples, in green, were only run at 2 dates. If we thought scan date was likely to affect the data, we would have to be concerned about it confounding our analyses of subtype. If we click the PCA plots link, we get plots showing the first 4 principal components of the data, colored by each covariate in the analysis. PCA condenses our 730-dimensional data into 4 maximally informative dimensions. In this plot, colored by subtype, we can see that the first 2 principal components very effectively separate all the subtypes from each other, and that the third principal component seems to separate the normal samples in green from the cancer samples. Now let's look at the same plot, but colored by binding density. We see little association of binding density with any of the first 3 principal components, but it seems to strongly drive the 4th principal component, seen here from this gradient of blue for low binding density to red for high binding density. This further illustrates the importance of adjusting for this technical variable in downstream analyses. Finally, by clicking on pathways on the left, we can see heat maps of each pathway's data. Here we see that the DNA repair pathway genes seem to cluster normal and subtype A samples apart from the rest of the tumors. Now let's look at the results of the pathway scoring analysis. Looking on the right, we see a heat map for the pathway scores in all the samples. Notice how the intractably fine-grained heat map of the 730 genes has shrunk to a much more intelligible 13 dimensions, 1 dimension for each pathway. On the left, a heat map shows a correlation matrix of the pathway scores. TGF-beta immediately stands out. It appears to be highly negatively correlated with the other pathways, but we will see later that the TGF-beta score likely has a different interpretation than the other scores. That this striking observation is possibly spurious should serve as a reminder that pathway scoring results must be interpreted very cautiously. By clicking on the PCA plots tab, we can see the first four principal components of the pathway scores. By clicking on the pathway names on the left of the screen, we can examine each pathway score in greater detail. The plot atop the screen shows the pathway scores within each subtype. Notice that the normal samples have lower apoptosis scores than the other subtypes. Since we've used normal samples as a baseline for pathway scoring, normals will have lower pathway scores on average than the other subtypes for every pathway. However, in this plot, the normal samples all fall very much to the extreme low end of the apoptosis scores, suggesting that the apoptosis pathway score reflects a continuum from normal to dysregulated pathway state. This pattern seems to be the case for many pathways. In contrast, when we look at the TGF-beta scores, we see that the normal samples fall close to the middle of the pathway score range. This pathway score clearly captures something different than the normal to dysregulated spectrum, explaining its lack of correlation with the other scores. Note that each pathway score has a different pattern across the subtypes. Here we see the apoptosis pathway score peaks in subtype B, whereas the cell cycle pathway score peaks in subtype C. Returning to the apoptosis pathway and scrolling down, we see the apoptosis pathway score plotted against the scores from other pathways. Looking at this plot, we can see that the apoptosis and Wnt pathway scores are highly correlated with each other. The plot of apoptosis versus DNA repair is more interesting. Apoptosis scores are higher in subtype B samples in blue, whereas DNA repair scores are higher in subtype C samples in red. We might speculate that these two subtypes take advantage of different oncogenic pathways to grow and survive. The apoptosis pathway's relationship with the TGF-beta pathway score is also interesting. The scores are highly correlated, but the odd behavior of the normal samples in the TGF-beta pathway seems to have reversed the pathway score's direction. Now let's look at the pathway level summary of these differential expression analyses. The pan-cancer pathways analysis module summarizes differential expression at the pathway level using two statistics, the global significance statistic and the directed global significance statistic. Global significance scores condense the differential expression results from 730 genes into 13 pathway-level measurements of differential expression. These simple statistics are calculated from the t-statistics of pathway genes, which are themselves calculated from linear regressions run in the differential expression analysis. Global significance statistics are calculated separately for each variable in the regression. The global significance statistic measures the cumulative evidence for the differential expression of genes in a pathway. It is calculated as the square root of the average squared t-statistic of a pathway's genes. The directed global significance statistic is similar in spirit to the global significance stat, but rather than measuring the tendency of a pathway to have differentially expressed genes, it measures the tendency of a pathway to have over or underexpressed genes. That is, it's directed. For each covariate, it is calculated as the square root of the average signed squared t-statistic. A pathway with both highly upregulated and highly downregulated genes can have a very high global significance statistic, but a directed global significance statistic that is relatively close to zero. The two statistics will be equal in a pathway that contains genes regulated in only one direction. Note that this approach is entirely unrelated to the mathematics behind the pathway scoring module. The pathway scoring module scores samples, whereas this module scores covariates. Here we see a heat map of global significance statistics for all pathways across all subtypes. Cell cycle genes in subtype C are particularly differentially expressed. On the whole, subtype C and subtype B seem to have the most extensive differential expression. And here we see a heat map of directed global significance scores. Here, highly upregulated pathways are in red and highly downregulated pathways are in blue. We can see that the differential expression in the cell cycle genes appears to be from upregulation. Whereas in most pathways, the average gene appears to be downregulated. The chromatin modification pathway is particularly interesting, as it is downregulated in subtypes B and A, but upregulated in subtypes C and D. We can also view the differential expression results for only a pathway's genes. Here, we'll look at the cell cycle pathway, and we'll look at the contrast between subtype C and normals. Here, the tendency for cell cycle genes to be overexpressed in subtype C is very apparent. At the bottom of the screen, we see the differential expression results for the top 10 genes in the cell cycle pathway. The genes CCNA2 and TTK are the most statistically significant cell cycle genes, And if we look at the volcano plot, we can see them atop the plot. Now let's look at the path view plots for the differential expression analyses. Navigating to the cell cycle pathway, we can see the biological context of differentially expressed genes. Genes that are upregulated compared to normal are colored red, and downregulated genes are green. Here, P53 appears to be mildly downregulated, while many of the genes in the CYC family appear to be strongly upregulated. As we look at the results from different subtypes, we see subtype-specific patterns of dysregulation. Subtype C's particularly extensive upregulation of cell cycle genes is apparent here. Note the mini-chromosome maintenance complex. This family of genes is highly upregulated in subtype C, but not at all upregulated in subtype A. Now let's look at the highest level analysis, pathway significance plots. These plots combine the results of pathway scoring analyses with pathway significance statistics calculated from the differential expression analysis. A plot is shown for each predictor variable in the differential expression analysis. Subtypes A, B, C, and D here. The horizontal axis summarizes the association between a pathway score and the variable. Essentially, we're rerunning the linear regressions from the differential expression analysis using pathway scores instead of genes. Each pathway's position on the horizontal axis shows its negative log 10 p-value. Simply put, pathways on the far right side of the plot have more statistically significant associations between their pathway scores and the variable in question. Here, the subtype A versus normal contrast. The vertical axis gives each pathway's global significance statistic. Pathways appearing at the top of the plot have the most extensive differential expression of their genes with the variable in question. Here, we see that only the notch and transcriptional misregulation pathways have impressive differences in their pathway scores between subtype A and normals. A different set of pathways, STAT, PI3 kinase, RAS, and TGF-beta, have the most differential expression at its single gene level. The unimpressive showing of these pathways on the horizontal axis indicates that the pathway scores for these pathways do not capture the subtype A versus normal difference. If we go to the plot for subtype C, we see much more impressive results. Note that the axes here extend to much greater numbers than they did for subtype A. The cell cycle pathway again stands out in both metrics. Since the cell cycle pathway is so impressive on both axes, we might conclude that the extensive differential expression in subtype C versus normal samples is driving a great deal of the variability in expression of cell cycle genes, at least enough to be captured in their first principal component. Thanks for listening, and I hope this was useful. If you have any questions, the Pan-Cancer Pathways Analysis Module white paper contains even more detailed discussion of how to set up your analysis and how to interpret your results.